Here is the underside of our little moth. I don't think Theo has seen this one, Claire. Of course, it's on a curved side of the sample, so I have to be ever so gentle. There it is. There are the eyes, and there is a little curvature of the mouth, and you can see the under part of the legs, the abdomen, the thorax here. Pretty remarkable that we can see some kind of detail like this, I think. Let's, maybe we'll take a picture like this, and let me see what I can do with the color correcting, see how much it'll change. That's kind of cool. I like that view too. What's really neat is you can see that little shimmering there is structural color. So it's not like actual pigment color, but rather it's the lighting and how the colors are reflected by the light in the exoskeleton, which I think is pretty neat. All right. Y'all let me know if y'all are interested in looking at anything else. And I mean, you ambers were so detailed to make it. Well, so turnside, it depends too on what kind of amber you get. So let me show you this one here that we have. This is not my favorite piece because there's a lot of junk in it, but it is 90 million years old. It was around during the time of dinosaurs, which kind of makes it a little bit extra special, I think. But it's full of it has a lot of schmutz in it too. So it's not as clean of a sample as these. Like normally, these ones that I've shown you, we have like a really, really nice, like it's optically very clear what we're looking at. It's see-through. Part of that is dependent on the, see, how do you know that the refraction and not the actual tissue pigmentation color, just from experience? Yeah, Fomkeen, by the way, Fomkeen, welcome in. So, a classic way of insects having color is structural color. So like beetles aren't pigmented. When you shine the light at them, it has almost this iridescence color where the different angle of light shows a different like coloring. And so that's the same thing that we see with this moth. So just like changing the light a little bit changes that color pattern. And that's what would be more that iridescence color would be structural color rather than pigment. Versus if we zoom in here, the wings on this individual, we didn't have, there's no changes in color. Like that's all pigment there. That's also a function of that tissue as well. So the wings are usually just a couple of layers of cells. And so they just don't have the room for structural color for those. Hopefully that makes sense, Fomkeen. But I did promise Turnside wants to see the sample that was around during the era of the dinosaurs. So I'm going to zoom out all the way first, and you guys can tell me what you see. Of course, Fomkeen. How are you doing tonight, my friend? I hope you're having a lovely evening. So let's zoom out all the way. So you see here there's a lot more schmutz here. There's just dirt. There's a bunch of other... There's insects. There's also something I think really, really cool. Actually, I think I had it on the wrong side. There we go. Yeah, so there's a whole lot going on in here. There's a couple of insects. There's a snail shell. The snail shell is my favorite on this because... Well, we'll zoom in and you'll see why. I think it's super, super exciting. By the way, where these samples come from, so most of our amber is 30 million years old. That is Dominican amber. Dominican amber is pretty readily available, and there's a couple of places where you can legally import and export them. So we do it through legal means. All the subs, bits on the channel, we put back into the stream, and my wife and I would do school visits. To teach kids about science. We give them stickers, as well as we leave a sample of amber with the classroom so the students can learn about the amazing life that happened 30 million years ago. This piece here is donated by a research lab to our stream with the assumption that we are not going to sell it or ever give it on. They had finished analyzing it for their own work, and they let us have it and utilize it. So we use it for talks and presentations and teaching people about the amazing biology of the world. So just a little aside there. So if we zoom in on here, we'll finish with the best part last, right? So we have a wasp-like creature as well as a termite. So the upper one is a termite right here. And then this lower one here is most likely a proto-wasp. So ants only evolved around 90 million years ago, so we'd probably be seeing some of the first ants in this. This is the very first ant discovered was 90 million years old, and it's called a hell ant. This is not it. The mouth is all wrong. So it's probably a proto-wasp. There is a termite above it, and actually let's go ahead and change the color. So the color looks a little bit less yellow. Boom! Look at that. Look at that. Already looks cleaner. If we go over here, there's a mite. It's not clear if this is like a parasitic mite or a food mite, but you can see it has a whole bunch of structure that you can see. Ant evolution from ant to wasp. Yes. Claire Burr and I were talking about this yesterday, about the possibility of if there is anything that's in reverse, and I couldn't think of any example of like that, of like kind of reversion to more wasp-like features. It seems like everything that's gone full ant has kind of stayed in that particular line versus like other kinds of reversions on that front. Only wasp to ant, no ant to wasp. Exactly. At least that's what the current knowledge says. But there's a little mite down there. If I had a gut call on it, I'd say it's parasitic because of the teeth. It's probably a blood-sucking mite or a tick. Those also evolved from like the same like phylogenetic tree or ancestral relatedness. So it's kind of hard to tell apart. We do need more fossils, as Professor Anduza said. That's a good command, Claire. I had no idea we had that command. Sometimes Claire adds some sneaky commands in, which I thoroughly appreciate because it means that we have like good quality content. She's the best mod ever, just so you know. Yes, am I throwing shades at other people's mods? Potentially. But Claire Burr makes it easy for us to live and function and do our thing. Alright. So let's zoom in. This is what I think is the coolest, and you guys will see why in a second. We've nicknamed this sample Gary. It is a snail. The digital zoom, you can see that the snail is still inside. What most likely happened is that when this sample got caught, it'd be nice not everyone can afford. I know, Claire. I can still be like, you know what? I got Claire bot. Wee-hoo! Love you, Claire. So you can see inside the sample there is that blue streakiness. And that blue in here, those little ruffle lines. Love you, Claire Burr. Those little ruffles are actually part of the snail's body. That's the part that like scuttles around its little feet. So what probably happened with this sample was that the amber flowed in, like into the shell, and then it just got caught. And so it's actually like totally preserved in there. We can't extract any DNA or RNA or nucleic acids or what have you from this sample. It's just too old to extract anything given current methods. We think everything is just totally degraded at this point. It's still pretty cool though. I water the plants in your happy place. That's amazing, Claire. That should totally be a thing. But yeah, pretty amazing, right, chat? At least I think so. I'm a sucker. I could also look at the samples like for all day. So that's why we have a dedicated microscopy stream, so that Valence rabbit holes and the need for looking at stuff under the scope can be satiated. Plus, I think everyone else also kind of enjoys the microscopy that we do